Szanowni Państwo, rozpoczynamy trzecią część naszego dzisiejszego spotkania i konferencji. Zapraszamy wszystkich Państwa z holu, prosimy o zajmowanie miejsc. Przed nami już za chwilę wykład pana profesora Benedikta Berningera. Pan profesor związany jest z Instytutem Chemii Fizjologicznej przy Uniwersyteckim Centrum Medycznym należącym do Uniwersytetu w Moguncji. Na Uniwersytecie Kalifornijskim w San Diego pan profesor badał krótkotrwałe oddziaływania neutrofin na zakończenia synaptyczne i wypustki neuronalne. Wykład, który pan profesor Berninger za chwilę wygłosi, dotyczyć będzie przemodelowania i przeprogramowania mózgu dorosłego człowieka. Po tym wykładzie zaproszę wszystkich prelegentów i naszych gości, naukowców z całego świata. Usiądziemy na tych fotelach i będziemy do Państwa dyspozycji, eksperci będą do Państwa dyspozycji. Będzie można zadać pytanie, podzielić się swoimi refleksjami, rozpocząć dyskusję na temat, który będzie Państwa pasjonował. Profesor Berninger.
Thank you very much for giving me the opportunity to speak here. I'm greatly honored that I was invited, and I have also to say I was greatly impressed by the presentations of this morning, so I hope that now with a switch to more basic neuroscience, I hope you will not get bored, but still bear with me. So in my presentation, I will discuss with you our attempts to both remodel and reprogram the adult brain. The adult brain, as we all are aware, of course, is a very vulnerable structure, and most of the times when neurons die, there's no way to replace them. This is an observation that has been already formulated more or less more than 100 years ago by Ramon y Cajal, when he stated that actually in the nervous system, everything dies, nothing regenerates. And he also said this, which is sometimes omitted, it's the signs of the future to change this verdict. So in a certain way, that's something we are, together with many other laboratories in the world, trying to do to change this verdict. So more practically, we are interested in essentially two types of questions. One is there refers to stem cells that exist in the brain. As you know, or as you probably all are sure, that in most parts of the brain there's no neurogenesis. There are some parts of the brain where neurogenesis occurs. And so these are of course not very widespread regions, but there is neurogenesis in some restricted regions. And of course it's very important to learn the principles of how actually these stem cells can generate neurons in a physiological process. So this is one of the topics that we are very interested, and this is just referring to the fact that there are stem cells residing in specific niches, and these cells can generate neurons. For example, in the human brain, it is believed that in the one subregion of the hippocampus called the dentate gyrus, even in human beings, neurogenesis is taking place on a daily basis, and even in people as they age. So it could be that at least in principle the human brain is capable of some neurogenesis. Now, of course, most parts of the brain aren't really equipped with these stem cells. So the region that probably is affected in most of the cases that we heard today, the cerebral cortex, is a region apparently devoid of stem cells. Now, when cells die there, what can we do? And it is actually the approach that we are taking is to harness the potential of other cells that are not neurons, and aiming at converting these cells into cells that then give rise to neurons. So I will talk a little bit about this approach, where we aim at converting glial cells, which are actually a very important populations of cells in our brains, to convert these glial cells in nerve cells. and I will tell you about the approaches that we have taken to do this. We try to learn as much as we can from those niches where neurogenesis occurs, because we believe that probably we can derive some principles of how actually neurogenesis could take place in an adult brain, and then see whether we can instruct cells in other parts of the brain to do something similar. I have to tell you, most of that, what we have been doing so far, is work in vitro. So this means in a dish. That's a very, it's already quite interesting and exciting for us. Of course, if you really want to move this towards another level and even think about moving at some stage such approaches to a clinical treatment, you need to show that these possibilities also work in an in vivo system. And I will talk about our approaches to indeed do this, to move from in vitro to in vivo. Now, finally, the big question is, of course, even if you are able to generate new neurons in a brain, how will these neurons integrate into the pre-existing network? So this shouldn't take place in a random fashion, but again, we probably have to believe that these regions where neurogenesis occurs can teach us lessons. And I will tell you a little bit about what we are doing there with respect to learning principles of how new neurons integrate into an adult brain. This will maybe teach us something of how we can then instruct cells to become neurons and to integrate and to take eventually also function. But it also gives us some ideas, actually, perhaps with respect to something which we heard a lot today about rehabilitation. So these are the two angles of integrating new neurons in a brain. One is, which I will talk towards the end of my presentation, refers to experience-dependent remodeling of the connectivity of neurons. And in the first part, I will discuss with you our attempts to reconstitute neurogenesis, or the generation of nerve cells in parts of the brain where this does not happen, the cerebral cortex. So there's a very important term that I will use throughout this presentation, which is reprogramming. You're maybe familiar with the great achievement of Shinya Yamanaka in 2006, being able to reprogram skin cells into pluripotent stem cells. That meant for the field that for the first time it was possible to derive from a patient essentially cells that can be giving rise to essentially all types of body cells, provided we have a strategy to derive from these pluripotent cells these more specific cell types that we have in our body. So we have been following something similar but a little bit different in a way that in Shinya's approach, what you have to do is you take a differentiated cell, you convert it into an entirely non-differentiated cell, and then you differentiate it back. So our approach, and this is something that I have been doing for many years together in collaboration with a very outstanding scientist in Germany, Magdalena Götz, we have been aiming at converting cells directly from one fate into another without going through this pluripotent state. So I will tell you exactly how we did this. I will use the term reprogramming in this sense, in this direct. I just want to point out that something that reprogramming is something conceptually quite remarkable, in the sense that when I was 15 years old, it was a knowledge that was clear that differentiated cells cannot be brought to another cell fate. So if you take a cell from the skin, it would be impossible to make a neuron out of this. It was thought to be totally irreversible. And I'm inspired by sometimes Greek mythology, which teaches us that things that seem impossible, like converting human beings into pigs, as depicted here, they seem impossible, but in some way, if we have a way to, a logical strategy to do this, it might not be impossible. Of course, conversion of humans into pigs is not very useful, and it's much more meaningful to do this approach, namely to try to convert glial cells into nerve cells. And the idea is that perhaps it would be possible to start with a glial cell that for sure has an important function, and we will not solve the problem without addressing the question, we cannot just lose these cells, we need them. But nonetheless, if we could entice these cells to give rise to the different kinds of cells that populate our brains, and specifically the great number of beautiful cells that populate our cerebral cortex. And so the approach that we have been taking is entirely experimental. It's not at a stage where I would say we can move this to the clinic, but particularly also because we use viruses at this moment. But this is, of course, something that we need to figure out first, and everything else can eventually then addressed by of course, the community. So the idea is to take viruses that can bring in specific genes that then convert this glial cell into a nerve cell. And the genes that we would like to bring in encode for transcription factors. So transcription factors are those factors that bind to the DNA, to the genome, and can there induce and regulate gene expression. And of course, a glial cell has another program of gene expression in a nerve cell. So the idea is simple. Can we use these powerful transcription factors to convert a glial cell program into a nerve cell program? And so there are several questions that we need to address. And this is maybe a very dry slide because it's sort of presents some milestones, but I think they are important to keep in mind. So one of the big questions is, if you follow this approach, what would be the best cell in the brain to be converted? And I have to tell you, at this stage, we are exploring all these possibilities. There are different glial cells in the brain, and perhaps one and the other lend itself more than the other cells to such a reprogramming. But I tell you what we have been doing so far. Then, of course, the question comes regarding the different possible strategies to convert the cell. So I have been telling you that we use essentially viruses, but of course, ultimately, other approaches might be interesting, which allow you to move this to a more clinical setting. Then, we of course would like to generate neurons that are true, not some fake neurons in the sense of they have some features of a neuron, but indeed they are not exactly matching what is in the brain. This is very difficult, but it's actually becoming more feasible because nowadays we understand better and better the molecular signature of each cell type in the brain. For example, in the cerebral cortex, pioneering work of my colleague Paula Lotter has been showing strategies that we can now really dissect the mechanisms that specify each type of neuron that is in the cerebral cortex. Then, when we do this in vitro, that the problem is solved. But in vivo, we have many other problems, namely the tissue in which this process should take place may provide some constraints and may actually not be very sort of helpful for this reprogramming process to take place. So we have to think about what is actually the environment, how the environment reacts on this process. And finally, of course, as I said, very crucial is the functional integration of these cells. So I tell you about the basic approach that we have taken early on, which was using, and most of the work that I show you has been performed in mouse as a model. I will also show you some data with human cells, but the focus of most of the work we have been doing so far is in mouse. In this case, we prepared cells from the cerebral cortex of early postnatal mice. These were 5 to 7 days old, cultured the cells in flasks, expanded them, and the cells that you get under these conditions are essentially so-called astrocytes. Astrocytes play a role as support cells, but they have also important functions in regulating the health of the neurons, but essentially they are, of course, not neurons themselves. You can culture these cells, and then what we did was we passaged these cells for some time, and then added these viruses that brought in transcription factors that could instruct a fate conversion, or that was our hope. And indeed, what we found was that when we did this with these astrocytes, they converted into neurons that also by electrophysiology could be demonstrated to have neuronal function. And what was also very exciting is that using different genes, we could convert the cells into different types of neurons. So here, neurons that use the neurotransmitter glutamate, but using another combination of factors, we could generate neurons that use the transmitter GABA. And this indicated to us, essentially, it should be possible to generate a subtype-specific arsenal of nerve cells. Now, it's something remarkable, of course, to move to the human cells. And I just show you this slide because I had to read a book in order to understand this painting or this fresco. I don't know, probably most of you have seen it, but not everybody probably has already recognized it, namely that this part is a sagittal section through a brain. So I was really impressed when I read this because I thought I had seen this so many times and had recognized what you can see there. But in fact, it's really impressive how the sagittal section is represented here. And what is true is that apparently Michelangelo used for his artwork, of course, his knowledge in human anatomy, and he must have seen dissections of human brains in order to have such a very good depiction of it. So what we did was much less artistic, but this is work that was conducted by Marisa Caro in my group, and what she did was to take cells that were isolated from brains of patients that had to undergo some surgery, and I don't know really, so I would have to ask you for the neurosurgical details because I'm a biologist. So we got the access channel, which meant that we didn't get the pathological tissue itself, but tissue that was sort of needed to be removed in order to get access to that. And we took this tissue and we cultured the cells, and what we found was that the cells that we obtained there, they were derived from those cells that are normally associated to blood vessels. These cells are called pericytes. These cells are very important of maintaining the barrier between the blood system and the brain, and in a normal situation, but upon injury, these cells can proliferate, so they start to divide and actually form often or contribute to the scar formation that can happen or happens when you have a brain injury. Something similar happens when you take these cells into culture, because then these cells also start to proliferate, and they express still a panel of markers that are classical for these pericytes. Now what we did is we tried our transcription factors and wanted to test whether these can convert these cells also into neurons, like the cells from the mouse. And so we tried one by one, but they all failed, until we started to think whether maybe combining factors could be working. And what we found was indeed that when we combined two factors, and it doesn't really matter which are the names, indeed what we found was that these cells could give rise to nerve cells. So this is just showing you some pictures that you get an idea. So we start with these kind of cells. They look very flat, a little bit fibroblast-like. They express markers that are characteristic of pericytes. When we put these two factors together using these viral vectors that, as I have shown you before, then we can convert these cells into cells that express markers of neurons, and also they really develop nice morphologies occasionally and can express neurotransmitters, like here the neurotransmitter GABA. Very importantly also, when we co-culture these cells with neurons from the mouse brain, so these cells now have a right environment to develop and mature, what we find is that these cells indeed actually are capable of producing electrical signals like action potentials, characteristic of neurons, and also they can be targeted by other neurons. So showing that these converted cells can give rise to neurons that have at least many of the features that are expected for a neuron. So I think this is a very important conclusion, because this means we do have cells in the brain that, at least under some conditions, can be brought to generate neurons. course, this is an in vitro process. So, so far. This is actually showing you how this process takes place, and unfortunately, I think I will not have the movie here. I forgot to actually put it on the same computer together. But it doesn't matter because you see here the stills from this movie. It's actually a very long movie. It takes about 2 minutes, and it shows you in a time lapse the sequence of a cell that starts here with this flat morphology to convert in the course of 2 weeks into a nerve cell. So this is a very slow process. This is not happening like a movement like you do like this, and it would be there, but it indicates that the cell, in order to be converted into a neuron, has to switch off presumably its own program and then activate an alternative program, that of a neuron. And exactly, we wanted to understand a little bit better what is happening in these early phases. So this goes a little bit into the details, and I hope you will not totally be bored at this stage with this part. But what we did is we put these viral vectors into these nerve cells, into these parasites, and then used reporters, so these cells become fluorescent, we can recognize them, and we used FACS sorting in order to get these cells back into a tube, and then determined the expression, the gene expression program in these cells. So this is simply showing you that when we sort these cells, you can very nicely get green cells, and here you see that some of these have already converted into nerve cells. Now, we did this, in this case, we did some transcriptome analysis, which means that we studied the gene expression profile, and we did this with material from 3 different patients. So when people do this, normally they, so transcriptome studies, one point is, of course, normally you want to do this with cells from one defined genetic background, because this helps you in the analysis. In this case, we have 3 different patient material, so every patient has a very different gene expression profile to start with. So it's not so easy to find statistically significant changes that happen. But what we found was that indeed this shows you the different treatments, and here we compared cells that were not transduced with the transcription factors, cells transduced with one transcription factor, so these are 2 different ones, and then the one that combines both. And what we found, that the one that combines both is the only one that shows this change in gene expression that is then consistent, as we found, with acquisition of an early neurogenic program. So this indicated that indeed these 2 factors together are capable of eliciting a gene expression program that is characteristic of cells while they undergo the conversion from a non-neuronal cell to a neuron. I'm not going to show you the details of this, but it's very important to state that this is very is very important, because sometimes in the studies you are concerned that you only have some features present, but not a real more systematic change of identity. And by this kind of analysis, you can really demonstrate that this takes place. And also it's very important to guide us to understand better, to generate very specific type of neurons. So this kind of study, of course, you could imagine, could be very interesting in first place for neurodegenerative diseases, where you have to replace one specific type of cell. But ultimately, we hope that by learning more the big principles, perhaps it will be possible also to use, to some extent, the self-organizational principles that developing tissues have, and that have been discovered to a large degree by Sasai. It would be very great if we can use these self-organizational principles and elicit a more sort of global regenerative process starting from some glial cells. Now, this was all in vitro. We moved from mouse to man. That's already very good. But now it would be really nice also to be able to be looking in vivo. And so this is work that has been conducted by Christoph Heinrich. And so the work, what he did was to make a lesion, an injury in a mouse brain. A very small traumatic lesion. So it's not, of course, as widespread as a real traumatic injury that happens in an accident. But nonetheless, there's a sort of like an area where there's a brain damage. And what he did now was, guided by the identification of factors that we have obtained in the in vitro study, he now asked the question, what happens if after some time, and this is relatively early here, so sometime when the cells in response to the injury start to proliferate, what happens if I bring in these two transcription factors, and asked whether this would also in vivo induce conversion of cells into neurons. And what you can see here is that if you put in a virus that does not bring in transcription factors, nothing happens. So here we stained for a marker of immature neurons, and we see no response. The same thing is true when we just applied one transcription factor alone, which is ACL1 in this case, and again, no response. So this tissue is not just by looking at it or by bringing in one factor allowing the conversion into neurons. However, when we introduced the two transcription factors that worked in our human experiment, what we found was that indeed, here you see the genesis of new neurons as illustrated by the staining for a immature neuron marker, double cortion. So these cells are on the way to become neurons. This is a very immature state, but this is remarkable enough that it can happen in an in vivo context. And we found actually cells that developed quite nice morphologies indicating that that it is also possible that these cells mature to a more differentiated state. Of course, this is at this stage highly inefficient process. Many cells remain in an immature state, and it will be a crucial question, of course, for us to figure out strategies to perform this conversion in a sort of more effective or efficient way, and also at the same time direct these cells towards specific neuronal identities. We then asked actually, what are these cells that give rise to these neurons? Because as I told you, our brain contains many types of glial cells, and so some cells might respond, others may not respond, and this is just showing that, and we will not have to go into the details here, but that indeed when we put in our viruses, we target many different types of glial cells. Some of them the astrocytes, some are cells that can give rise to oligodendrocytes, which are the myelinating cells, and also microglia, which is a kind of immune cell of the brain. Now, using some genetic tools, we could demonstrate that it is actually very specifically one cell type that I have not been discussing with you before, the so-called Ng2 glia. This is a glial cell population also that exists also in our brains, which with a very low pace proliferates even in the adult brain. And these glial cells, they respond to the injury. They also they accelerate the proliferation, and when transduced with these vectors that bring in the transcription factors, these cells can undergo conversion into immature neurons. We found, and this is a little bit puzzling, and we don't understand this yet, that this effect in vivo in the adult cerebral cortex seems to be dependent on the prior injury. If we did the same experiment with a little different conditions, but without an injury, we did not see this neurogenesis. So it could be that the injury exerts an effect on the glial cell population, and by this renders these cells more permissive to our strategy. So we don't understand what's really happening there, and of course this is a critical question to be addressed. So, but it indicates that these factors are not the whole story. There are more components to be considered if you really want to understand this conversion process in the brain. So we also addressed the question whether these cells in vivo acquire some neuronal properties. And again, I have to say, we are at a very early stage, so we found that cells can exhibit electrical properties of neurons, and we can record these cells in brain slices, and we find that yes, these cells do receive, they are excitable, and also that these cells can receive input from other neurons, indicating that while they convert into this, in the brain in vivo, these cells can be targeted by neurons surrounding them and sort of integrate into the network. But we have to still understand whether this is something they inherited from their ancestor, the NG2 glia, or whether these connections are really newly de novo formed synapses that are generated as these cells converted. So there are many open questions, and this is something that we will have to do in the next probably decades. Now, we also wanted to know understand a little bit better what is the importance of the injury. And one idea was that perhaps the injury simply is important because cells proliferate more strongly right after the injury. So we thought if that's the case, then if we move this whole thing into a postnatal stage where glial cells are still proliferating in a mouse brain, perhaps at that stage we don't need the injury in order to cause the conversion. And so what we did here was to make experiments in very young mice, but at a postnatal stage, and then address the question whether without causing a particular injury before, just by introducing the viruses, we can cause conversion. And this is work that has been done by Sophie Perron in my lab, and she found that indeed, just bringing in these vectors into the postnatal brain, so this early into mouse brains of 5 to 7 day old mice. What you found was that indeed you can see here conversion, and what is very beautiful in this picture, it shows you brother and sister. So one of these cells did not convert, while the other cell converted into a double coating positive immature young neuron, which shows these two sides that, or it shows nicely that actually this cell originated from a glial cell, but the process is not 100% efficient. In this case, it's 50% efficient. So it shows you that of course we have to learn more how to do this in a more effective way. Okay, I'm not going to read these conclusions to you for this part. It's simply, I think I hope I have shown you how we are pursuing this field, and sort of this shows you the general idea that it would be possible to force cells in the injury to convert into neurons by inducing a neurogenic program using transcription factors. This very complicated slide only shows that we are not the only ones doing this nowadays. There are many labs in the world that investigate this, and there are some remarkable things also observed in, for example, certain types of injury. Work from a Swedish group, the laboratory of Jonas Frisén at Karolinska Institute, but also an Italian group, Federico Luzzatti from the University of Torino, have shown that in the striatum, at least of mice, if you cause a stroke injury, some cells spontaneously, without having to be receiving any transcription factors, can give rise to immature neurons. And they undergo a sequence of steps where they first divide and then acquire neuronal or immature neural properties and then can differentiate into neurons. The problem is that many of these neurons die, so they don't manage to establish themselves in a stable way. And of course, then the question is, what can we do to render this more effective? So our approach has been using transcription factors, and again, other groups pursue this pathway and have found that also the astrocytes can be reprogrammed. So they have taken other tools, other strategies, but they have found that also astrocytes can be converted both in the striatum as well as the cerebral cortex. So this indicates that we need to, or I want to point out that we need to learn the logic of this process in different parts of the brain. So it's probably something that has to be adapted to wherever tissue in the brain, in the CNS, you look at. Good. So this is part one. I don't know how I'm in time. I'm still
5 minutes.
5 minutes? Okay, great. So I just wanted to pick up another interesting topic, and I will be quick with that, which may be interesting also in the context of when we think about rehabilitation. And this actually refers to the fact that we do have, even we humans do have neurogenesis in some parts of our brains. And one part that we are particularly interested in is the so-called dentate gyrus of the hippocampus. That's a subfield in the hippocampus, a subregion in the hippocampus. And hippocampus is a region that is very important for our memory function, but also regulation of emotions. So this is a very important structure, and there are neurons that are generated lifelong. Now, the question, and they are generated from stem cells that you can see here, and these cells divide and can give rise to beautiful granule neurons. As these granule neurons mature from an immature cell, as you see here, they also develop their contacts. And we were very interested how actually these contacts develop over time. So this is not happening all overnight, but over the progress of weeks, and probably in humans over the progress of months. And the question would be, can we modify, or can this sort of like this becoming connected to the network be modified by experience? And so the way that we did was, and maybe I skipped, this is a technical slide, the idea was to use a paradigm to study whether connections can be altered, a paradigm that has been developed by a very important laboratory in the stem cell field, by the laboratory of Rusty Gage. And this is shown here, illustrating you, and maybe the most important thing are these two panels here, and the comparisons, the control here, where they found that placing mice in an enriched environment or allowing these mice to do exercise, more neurons were born. So there are more neurons born, but also more neurons survive and integrate. So imagine, or actually during this process, many more neurons are born, and normally a quite large percentage of cells dies again. But when these mice were exposed to certain environmental conditions, more of these cells survived. And we addressed the questions, and this was a work by Matteo Bergami, the question whether this also, we can see whether this has any impact on the connections these neurons receive. And so what Matteo did was he placed mice either in this boring standard housing or in this more interesting looking environment. And what he did then was using a viral method to trace the connection. So what we can do is we can look at one cell or at a cell population, which we call starter cell population, and see which cells are making synapses and how many cells make synapses on this starter population. And this is maybe the key slide for this part of the talk. Is what we did here, we looked at the connectivity of newborn neurons in the adult hippocampus. And we compared, we always looked at the effect that this exposure to an enriched environment had with respect to the birth date of this neuron. And this is the conclusion that can be drawn from this experiment is the following, and I'll show you the data in a second. It's very important that the experience is made during a critical period of the life of the neuron. If the neuron is born, but at this very moment you put the mouse in the enriched environment, the effect of the enriched environment is not very strong. The effect is really critical only 2 weeks after this neuron was born. So this indicates the neuron, as it matures, enters into a critical phase where it needs stimulation. Then this has a very strong impact and its connectivity is changed. And this you see here. This is one condition. I'm not going into the detail. You don't see very big differences. But in this paradigm, in this condition where we hit this critical window, where sort of like this window of opportunity, the difference is very remarkable. You see here many less green cells than here, and this indicates these cells here where the mice were exposed to an enriched environment, but at a given point after the birth of these new neurons, there's a remarkable effect. So I'm skipping this for the sake of time. These are only details illustrating how indeed this is a robust phenomenon. We can look at different connections in the brain, connections from, for example, one major input arises from the entorhinal cortex to the hippocampus, and you see how this enriched environment has a robust effect on connectivity. We can label many more cells in the entorhinal cortex when we expose mice to enriched environment. This is also reflected in the morphology of these neurons. So when we expose them to enriched environment at the right moment, this results in an enhancement of these structures, which are synaptic spines, which are the points where synapses contact these newborn neurons, indicating that indeed there is increased synaptogenesis happening if these newborn neurons are hit by experience during the right moment of their ontogeny. So what we identified was then a critical period where we can modify, where the connectivity of newborn neurons in the adult brain are strongly impacted. Of course, we have newborn neurons generated every moment, so every moment with respect to this system, some cells are in the critical period, while others have not yet reached it and others already left it. But this indicates that when we also think about rehabilitation, of course, there might be particular windows of opportunity with respect to the nerve cells and the other cells. And so this is just illustrating us some of the logic that we have to keep in mind, how cells integrate into a brain. So this is giving you a long list of all the collaborators that have been working over the last year. So I want to point out Magdalena Götz from the Helmholtz Center in Munich. We have a very fruitful collaboration with Klaus Konzelmann for the connectivity. I have a very good friend in Argentina with whom we have been working on the adult neurogenesis project, Alejandro Schinder. And the people who did the work in my laboratory were Marisa Cardo, most important, Matteo Bergami, and Christoph Heinrich for the presentation that I gave you today. And I'm happy to receive some more questions.
Thank you very much.